Once again, good morning everyone. This is the continuation of our discussion. I am Jefferson Milante and I am assigned to discuss to you about the requirement for an infectious dose and attaching to the host phase 2. To guide you class on my discussion, here's my objective. At the end of my discussion, you should be able to explain what is meant by the infectious dose using examples and describe the process of adhesion and various mechanisms by which microbes use it to gain entry. Now let's proceed what is meant by the infectious dose or ID. Major factors in the development of an infection, the requirement for an infectious dose. The infectious dose or ID refers to the minimum number of microbial cells required to initiate infection in the host. The ID varies widely among microbial species. Microorganisms with smaller infectious doses have greater virulence. External conditions, example stomach acidity, can significantly impact the effective ID. Vibrio cholerae or cholera requires a much higher ID than other pathogens due to its sensitivity to stomach acid. So, the infectious dose is the minimum number of germs na kailangan para magkaroon ka ng sakit. Ang infectious dose varies greatly depending on the type of germ. Mayroon tayong germs na kapag lower infectious dose are more dangerous because fewer are needed to cause an infection. External factors tulad ng stomach acid can also affect the infectious dose. Some germs are more easily killed by stomach acid than others. This is the table about the estimated infectious doses of selected pathogens. Like for example, the agent of measles. Infectious dose estimate 1 virus. Primary route of infection, respiratory. So sa measles, ang 1 virus is enough to make you sick. And you catch it through breathing in the air. And another example sa cholera. Infectious dose estimate 100 millions of cells. Primary route of infection, ingestion. Sa cholera requires a much larger number of germs, which is about 100 millions cells, and is spread through eating or drinking contaminated food or water. Ayan, the table shows that the number of germs needed varies widely depending on the disease. Mayroon din tayong agent of Q fever, about 1 to 10 cells. Primary route of infection, respiratory. Ang Q fever, a bacterial infection spread through contact with animals or their waste. And another agent of tularemia, about 10 to 50 cells. Primary route of infection, virus. Ang tularemia, a bacterial infection that can affect many parts of the body. It's spread by ticks, fleas, and other insects. And another agent of smallpox, about 10 to 100 viruses. Ang primary route of infection, respiratory. Ang smallpox, a serious viral infection that has been eradicated globally through vaccination. Ang brucellosis naman is about 10 to 100 cells. Ang primary route of infection is virus. Ang brucellosis, a bacterial infection spread through contact with infected animals or their products. Another agent of viral encephalitis. Infectious dose estimate 10 to 100 viruses. Ang primary route of infection is mosquito bite. Ang viral encephalitis, inflammation of the brain caused by a virus, often transmitted by mosquitoes siya. And ang phleg naman is about 100 to 500 cells. Primary route of infection, phlegabite. Ang phleg naman is a serious bacteria infection spread mostly by flies and live on rodents. Ang gonorrhea naman, a sexually transmitted bacterial infection. And about 1,000 cells. Primary route of infection, sexual contact. Ang anthrax naman, a serious bacterial infection siya that can be contracted through skin contact, inhalation, or ingestion. It is about 8,000 to 50,000 spores. Primary route of infection, respiratory, cutaneous. Agent of typhoid, 10,000 cells. Primary route of infection, ingestion. Ang typhoid is a bacterial infection spread through contaminated food and water. Ayan, last agent of cholera na mention na natin a while ago. Now, let's proceed to the role of animal and human studies in understanding infection. For the first bullet, in 1900, Dr. Jesse Lazier, a Cuban physician working with Walter Reed, deliberately exposed himself and two volunteers to yellow fever infected mosquitoes to prove their role in transmission. While all three contracted the disease, Lazier died from yellow fever as a result of this experiment. So, it describes an experiment in 1900 where a doctor and two volunteers intentionally let themselves be bitten by mosquitoes carrying yellow fever to prove that mosquitoes spread the disease, and one of them died. Yan. For the second bullet, between 1954 and 1973, the US government conducted 153 tests, Operation White Coat, exposing approximately 2,000 Seventh-day Adventists, conscientious objectors to biological warfare agents to study their infectivity. While some highly virulent pathogens were used, the deadliest agents, anthrax, plugs, smallpox, were not. Ayan, it mentioned a US government study, Operation White Coat, between 1954 and 1973. Around 2,000 people who refused military service for religious reasons volunteered to be exposed to different germs to see how infectious they were. The deadliest germs, yan, the anthrax, fleas, and smallpox, were not used to this study. Now, let's proceed to the attaching to the host, phase 2. How pathogens attach. We have adhesion. Adhesion is a process by which microbes gain a more stable foothold at the portal of entry. Yan. Adhesion, this is the process by which germs get a firm grip on the body's entry point like the nose, mouth, or a cut. And bacterial, fungal, and protozoal pathogens attach most often by appendages and surface structures such as fimbriae, pili, flagella, and adhesive slimes or capsules. Viruses attach by means of specialized receptors. So ang bacteria fungi and protozoa, those are the single cells organisms na nagagamit ng virus structures like tiny hairs, which is the fimbriae or pili, whip-like tails, flagella, or sticky coatings, which is the capsules, to stick to the body. Viruses use special molecules, receptors, to attach the specific cells in the body. Mechanisms, fimbriae or pili, hair-like appendages that mediate attachment. Ang fimbriae or pili, these are hair-like structure on the bacteria that help them attach. Flagella, although primarily for motility, some bacteria use flagella for attachment. So, sa flagella, although mainly used for movement, some bacteria use flagella to help them attach. And sa capsules naman, polysaccharide layers that enhance adhesion and protect against host defense. Capsules, these are sugary outer layers that help bacteria stick and protect themselves from the body defenses. And sa viral receptors naman, viruses attach to specific host cell receptors. Viral receptors, it is a viruses have molecules that precisely match molecules on the surface of our cells, allowing them to dock. And sa hooks or suckers, parasites, mechanical attachment used by parasitic. Sa suckers or hooks, some parasitic organisms use hooks or suckers to attach mechanically. This figures shows three ways germs attach to cells in the body. Ayan, fimbriae. Bacteria use tiny hair-like structures called fimbriae to stick to the surface of a host cell. The image shows the fimbriae acting like hooks grabbing onto the cell. And sa B, capsules, some bacteria have a sticky outer layer called a capsule. This capsule helps them adhere to the host cell surface. The image shows the capsule acting like glue, holding in the bacteria to the cell. And the last one is the spikes. Viruses attach to the host cells using spikes on their outer surface. These spikes fit into specific receptors on the host cell, like a key fitting into a block. The image shows the viral spikes binding to the host cell. The figure demonstrates the different mechanism pathogens used to firmly attach themselves to host cells, a crucial step in infection. This table shows about the adhesion properties of microbes. It shows how different types of germs or microbes attach to the body to cause various diseases. Microbe, we have Neisseria gonorrhea. Disease, gonorrhea. Adhesion mechanisms, fimbriae attached to genital epithelium. So fimbriae are tiny hairs stick to the genital area. Sa Escherichia coli naman, disease, diarrhea. Adhesion mechanism will develop fimbriae adhescent. Special molecules on its fimbriae help it stick. Sa Shigella and Salmonella, disease, gastroenteritis. Adhesion mechanism, fimbriae can attach to intestinal epithelium. So fimbriae help it attach to the intestines. Vibrio disease, cholera. Adhesion mechanisms. Glycocalyx anchors microbes to intestinal epithelium. A sticky outer layer, which is the glycocalyx, anchors it to the intestines. Treponema syphilis, tapered hook embeds in host. A hook-like structure embeds itself in host cells. Sa Mycoplasma disease, pneumonia, specialized tip at ends of bacteria fuses tightly to lung epithelium. Pseudomonas aeruginosa, burn, lung infectious, fimbriae and slime layer. We also have microbe Streptococcus mutans or S. sobrinus. Disease, dental caries. Adhesion mechanism, dextran slime layer glues cocci to tooth surface. Influenza virus disease, influenza. Adhesion mechanisms, viral spikes react with receptors on respiratory surface. Foliovirus disease, polio. Adhesion mechanism, capsid proteins attached to receptors on susceptible cells. Microbe HIV, and disease nyo is AIDS. Adhesion mechanism, viral spike adhere to white blood cell receptors. Ang Giardia lamblia protozoan. This is giardiasis. Adhesion mechanism, small suction disc on underside attaches to intestinal surface. And the last is trepa- trypanosoma or protozoan. This is African and S. American trypanosomiasis. Ang adhesion mechanism, flagellum is needed to penetrate and stay alive. Here's the key takeaways that you need to remember. Infectious dose or ID. It is the minimum number of microbes needed to cause infection. So this is the smallest number of germs needed to cause an infection. And smaller ID equals greater virulence. So it means microbes with smaller IDs are more likely to cause disease. Sa environmental factors naman, conditions like stomach acid can impact the infectious dose required. Adhesion, the process by which microbes attach to host cells. So adhesion, this is the process of germs sticking to the body's cells. Specificity, microbes attach to specific receptors on host cells. So meaning, ang germs only attach to specific types of body cells. Attachment methods, Fabry, flagella, capsules, receptors, hooks, and suckers are all used by microbes to adhere. So, germs use various methods to attach, including tiny hairs, tails, sticky coatings, hooks, and suckers. Sa ethical consideration, historical human experimentation advanced our understanding of infectious diseases, but raised significant ethical concerns, leading to stricter guidelines and informed consent in modern research. Understanding infectious dose, adhesion, and invasion is very important for developing effective treatments and preventive measures against microbial diseases. That's all, guys, and thank you for listening.